And finally, one more video that deals with molecular movement. We've looked at diffusion, we've considered osmosis. Let's look at one final set of slides here dealing with molecular movement. Here we're dealing with active transport. Active transport is the process where we're going to move something from a lower to a higher concentration. There's a concentration difference, yes. Here you have a proton, but we're going to move it from an area of low, not many of these, to an area of high, where we have a lot of them. And as you can imagine, this is going to require some energy. We're going against the gradient. If you are trying to go into the movie theater when everyone's leaving the movie theater, you're trying to go against the gradient and you're trying to push your way in maybe, where the force is kind of being pushed towards you. You're going to need to invest some energy if you want to go against the gradient. And if this cell wants to take this thing, this proton, and put it out here with the rest of these, it's going to require some energy, and that's where ATP comes in. We've not spent much time with ATP yet. We're about to. But ATP is adenosine triphosphate, and basically it's the currency, it's the energy currency, how our cells operate. It's basically a unit of energy they can use to make something happen. In this case, it's going to change this protein's shape to allow the movement of this particular particle from here to there. So active transport is going to be important for two reasons. It's going to require energy, and we're going against the concentration gradient, different than passive transport like diffusion. And we'll draw that distinction more in a second. Here's another example. Here we're moving potassium, and again, we want to move it from a low concentration where we have very little to a high concentration. Potassium wants to leave the cell because there's lots of these things bouncing off of one another, and we're going to force this potassium into the cell. But to do that, we're going to have to spend some energy, which causes, let me erase this. Notice the shape of this protein, this gray structure, how it kind of flips like this. That requires energy, and that requires ATP. So that's why active transport is different, because of the energy need. So just some examples of this, and then we'll do one more example of molecular movement. So there are many cases where we want to push ions or push minerals into a high concentration area. We want to concentrate them for some particular purpose. Here you see metal ions like sodium and potassium, magnesium, and calcium require us to push ions to have them in a high kind of holding area to make some process about to happen. One example of that is the sodium-potassium pump. This is going to be critical for making sure your nerves work. We're going to put a lot of sodium on the outside of the cell and a lot of potassium on the inside. This pump you see here, sodium-potassium pump, refers to this protein. If we spend ATP energy, we can change the shape of that protein and push sodium out, push potassium in, and therefore we have this huge concentration difference, which is really critical right before your nerve fires, right before it sends a signal. So we end up with a high concentration of sodium on one side and a high concentration of potassium on the other. The only way we get that is by active transport. How do we maintain that concentration difference? Is sodium can't go through the cell membrane because it's not going to be a lipid. Potassium can't pass through the membrane because it can't go back out. So we end up with this concentration difference. Really important. Urinary system, another example where we see active transport, and we'll focus on that more in AMP2. So just to compare these just for a second, passive transport, like we've already mentioned, if you think about diffusion or think about facilitated diffusion, we're going from a high concentration to a low concentration, and energy is not required. Active transport, we're going from a lower concentration to a higher concentration, and energy is required. Okay. Just to wrap up here, I'll give you another case of molecular movement that's a little bit different. This is referred to as vesicular movement. Hard to say this morning. It involves vesicles. We're going to transport through the membrane, not through a channel, but through the membrane itself. Kind of like knocking a hole in the wall to bring something in, or knocking a hole in the wall to bring something out. So here's an example. Endocytosis is when we bring in a particle into the membrane itself. And here you see three kinds. You see phagocytosis, pinocytosis, and receptor-mediated endocytosis. What that refers to is, in phagocytosis, that's kind of like cell eating. Notice what happens to this particle. It comes to the cell membrane and it's engulfed. We basically are going to wrap the cell membrane around it, kind of like pushing your finger into a balloon, how the balloon kind of wraps around your finger. We end up with this vesicle inside of the cell made of membrane that now contains that particle. So phagocytosis refers to cell eating. Pinocytosis refers to cell drinking. Here we're going to bring in fluid. And again, once again, we have this vesicle. It's made of membrane. It's made of this stuff that just got pinched off. You see it being pinched off here. It gets pinched off, and now we have some fluid inside a membrane-bound container that is pinocytosis, or cell drinking. Receptor-mediated means that we have these little protein markers on the cell membrane surface. And if one of these stars, for example, gets into this protein marker, it's like turning a key. That tells the cell membrane, ooh, we now need to pinch that off and grab that. So receptor-mediated is a signaled form of endocytosis. The reverse of that is exocytosis. When you see the term exo, or ex, that means to leave or to go out, like an exit. Here, the cell has made something, perhaps these are protein particles of some kind, and this vesicle, this membrane-bound container, is going to fuse with the cell membrane, and when it does, it kind of opens up, and these particles are all going to leave, kind of like putting two bubbles in the bath kind of join together and make one big bubble. In this case, once we join the bubbles, we're going to exit out the particles that were in there, and that's exocytosis. So just looking at this slide, and here you may want to pause it and look at this in more detail because it's kind of busy. Let me get out of the way here. But you're seeing both exo and endocytosis. So over here is exocytosis. Okay, we're having the exit of these particles. Over here is showing you some endocytosis. We're ingesting this substance, and once we get it into the cell in a vesicle, then we can actually join it with a lysosome, and you see here we can then digest it once we bring it in. Here's phagocytosis. We've brought in some particle. We're going to break it down, and here's receptor-mediated, because that has the receptors, the little proteins on the surface that tells the cell membrane to fold around that object. Okay, some examples quickly here. Phagocytosis is a very common process for white blood cells. Our white blood cells are going to be looking for any foreign invaders. So if they find a bacteria or a virus, they may just engulf it and bring it in and destroy it. Exocytosis is going to be very important as the cell produces things like proteins or hormones. How does the hormone get out of a cell? It has to be exported out through exocytosis. Okay, and finally, why do all these things matter? Why does it matter how ions are moving or why we have these processes? It's really important for a couple of reasons. One, homeostasis depends on proper water ion distributions. There are sort of three compartments, if you will, inside your body that are going to have water. We talk about the water that's in your plasma. We talk about the water that's between your cells. This is called interstitial fluid, and the water that's in your cells themselves. So it is uber important that we have the right concentrations of stuff in all of these locations. If we had all of a sudden a lot of stuff, a lot of solute inside of a cell, that's going to draw water into this cell. By what process? Osmosis, because the cell is founding itself in a hypotonic solution. Flip side, if all of a sudden there's a lot of stuff in here, let's say someone eats a big bag of chips that's salty, okay, well now you're going to actually draw water from your cells into this, and you're going to have a lot of water in your bloodstream, and that's going to increase your pressure, which is what we know people who have high sodium diets have high pressure. It's because of homeostasis and water ion movement because of osmosis. The other example I'll point out to you is one we kind of talked about with the nervous system is if you have the right movement of ions, ions are going to help us generate electric charge if we have the right distributions on either side of a cell membrane. If I have a lot of ions on one side and not as many on the other, I have a charge difference in addition to an ion difference, and that's important for things like the nervous system to set up the potential for electricity to occur, movement of ions to occur through the cell membrane. So please, when you go back through these videos looking at diffusion, facilitated diffusion, active transport, osmosis, endo and exocytosis, these are all very important at a very basic level so that we have water and ions in the proper locations. That does it for this video. Thanks.